We all know mRNA vaccines were one of the biggest scientific wins of our lifetime. I mean, they completely changed the game in the fight against COVID-19. But what if that was just the warm-up act? Today, we're going to dive into a brand new breakthrough that promises to make these incredible vaccines even more powerful. Let's get into it.
So, this is the big question, right? It's exactly what researchers at Peking University were asking. They saw how amazing mRNA vaccines were, but they also saw these, let's call them areas for improvement. They figured they could overcome the core limitations and really supercharge their effectiveness.
Okay, so before we can really appreciate the solution, we've got to understand the problem. What are the challenges that are kind of holding back our current mRNA technology?
Well, it really comes down to this core trade-off. On one hand, these vaccines are fantastic. They're safe, and you can design and produce them incredibly fast. But on the other hand, the mRNA molecule itself, it's notoriously fragile, it's unstable, and it's not always super efficient at its main job, which is telling our cells to make the protein that kicks our immune system into gear.
To really get how scientists cracked this problem, we need to take a little trip. We're going to shrink ourselves down, way down, and go inside one of our cells to see its amazing protein-making factory in action. The first thing you'd see in this tiny factory is the messenger RNA, or mRNA for short. The easiest way to think of this is as the architectural blueprint. It's just a long string of code that carries the exact instructions for building one specific protein, like the spike protein from a virus. So that blueprint gets fed into the ribosome. And the ribosome, that's the factory's automated assembly line. It just chugs along the mRNA blueprint, reading the instructions, and it starts building the protein, piece by piece. But where does the assembly line get its parts, the raw materials? That's where the hero of our story comes in, transfer RNA, or tRNA. Think of tRNAs as a whole fleet of tiny, specialized delivery trucks. Each type of truck has one job. Find one specific building block, an amino acid, and bring it to the ribosome when the blueprint calls for it. And this, right here, is where the bottleneck happens. It's a classic supply chain problem. If a blueprint from a virus is demanding a huge amount of one or two specific parts, but the cell just doesn't have enough of the right tRNA delivery trucks, you get a traffic jam. The whole assembly line just grinds to a halt, or at least slows way down. And that's what limits how much protein gets made. So if a shortage of specific delivery trucks is the problem, the solution is actually beautifully simple, isn't it? Just get more trucks. And that's exactly what researchers did with a strategy they called tRNA+. Instead of trying to rewrite the mRNA blueprint, they just asked, why not give the factory more of the delivery trucks it needs? The tRNA+ strategy is literally that. You artificially give the cell a flood of the specific tRNAs that are in high demand, and boom, you break the bottleneck. And the result? It was pretty staggering. In their lab tests, just adding the right tRNAs boosted protein production by up to 4.7 times. You can really see the difference here. With a standard mRNA blueprint, protein production is, at a certain level. But when they added the tRNA+ strategy, that output just gets massively amplified. The researchers just gave the cellular factory the tools it was screaming for. But flooding the factory with more trucks was just the beginning. The researchers' next idea was even cooler. They started thinking, what if we could upgrade the trucks themselves? So yeah, that was the next question they asked. They knew quantity worked wonders, but what about quality? Could they actually chemically engineer the tRNA molecules to make them fundamentally better at their jobs? Faster, stronger, maybe even stealthier? And the answer was a huge yes. By adding some specific chemical tweaks to these synthetic tRNAs, they gave them some serious performance upgrades. They made them faster at reading the mRNA blueprint, they stabilized the molecule, making it more durable, and, this part is crucial, some modifications made the tRNA stealthier, so it was less likely to trigger an unwanted immune reaction in the cell.
Okay, so this is where it all comes together. The team took their quantity strategy, the tRNA+, and combined it with their quality strategy, these new supercharged tRNA molecules, to create a truly next-generation vaccine. Then it was time to test it in mice. The experiment itself was pretty straightforward. They took the standard spike protein mRNA, but this time, when they packaged it into the vaccine's delivery vehicle, a lipid nanoparticle, they also put in their new chemically modified supercharged tRNA molecules. Then they immunized mice with this new combo vaccine.
And the result in a living system was just undeniable. The mice that got the new vaccine produced 2.52 times more virus-neutralizing antibodies than the mice that got the standard vaccine.
So this wasn't just about making more protein in a lab dish. It translated directly to a significantly more powerful, more protective immune response in a living animal. The supercharged vaccine basically prompted the body to create a much bigger, stronger army of antibodies.
So what's the big takeaway here? The tRNA plus strategy really delivered. It boosted not only the antibody response, that's the body's search and destroy patrol, but also its cellular immunity, the T cell response that hunts down already infected cells. This really isn't just a small upgrade. It's a potential blueprint for a whole new generation of mRNA medicines.
And that just leaves us with this final, really exciting thought. This research was focused on a vaccine for an infectious disease, but this whole strategy of optimizing the body's own protein factory could theoretically work for anything based on mRNA. We're talking cancer therapies, treatments for rare genetic diseases. We may have just seen the door get kicked open to a whole new world of medicine.